All right. Well, good morning, early afternoon to everybody out in the Asia Pacific area. It's really great to be here today. And like Sierra mentioned, we're going to be really diving into getting micro C going in your lab, really from the pipetting portion of it all the way through to building contact matrices. So I'm not going to spend too much time on the outline. Sierra covered that for us already. So let's get into what exactly is micro C and what can it do. Um, so first, let's talk about the core workflow here. For those who have done proximity ligation work like Hi-C before, should seem very similar to you. You start with cross-linked cells, then that chromatin is fragmented. Those free ends are then ligated back as a proportion of three-dimensional space. And then those crosslinks are reversed, DNA is purified, and then you make a sequencing library. And so the differences between micro C and Hi-C really come in to play in a few different areas. So I'm showing the difference in micro C on the top compared to Hi-C at the bottom. So for micro C, we use a dual crosslinker, which better stabilizes the chromatin interactions. As opposed to a single cross-linker formaldehyde, which is used in Hi-C. And so our dual cross-linker does a few things for us. One, makes the chromatin structure a bit more stable. And it does that by binding proteins to proteins, which actually helps us enrich for that long-range information. And then it also keeps the molecules quite stable during the digestion. Now, when we digest the chromatin or fragment it, we use the imidase enzyme as opposed to restriction enzymes. And we're going to go into quite a bit of depth about this and why this makes micro C so much better than just using a restriction enzyme approach. Then the next difference really comes into play at the ligation. So when you have a restriction enzyme-based digestion, you know exactly what base is hanging out on that free end because you know you cut out a motif. With the imidase fragmentation, you don't really know what base is going to be at the end of these free ends. So we have a two-step bridge-mediated ligation. The biotin is captured on this internal custom oligo bridge sequence. Then we reverse cross links like normal, and then we do a biotin pull down and library conversion like you would with a Hi-C library. So I kind of covered this in a bit more detail in the previous slide, but just to kind of hammer this home, dual crosslinkers really help stabilize the chromatin, which enriches for long-range interactions. The imidase fragmentation to mono, di, and trinucleosomes, as opposed to a motif-dependent, gives us higher resolution, a nucleosome resolution, and improved genomic coverage. So we get a lot of questions around what happens in nucleosome-free regions. Well, we don't digest all the way to mono nucleosomes. We have a mixture of di and tri, so that linker DNA is preserved at some level. So no matter what, you're always going to have some fraction of the genome being covered, even in a nucleosome-free region. Then again, this two-step bridge ligation event is mediated because of those random bases. And again, that's really the only way to get around that randomness of the fragmentation to really get down to that nucleosome level resolution. Again, crosslinks are reversed, same as normal, right? Proteinase K, choose away the proteins. Now you have DNA that you want to purify away from those proteins. And then you convert into a sequence library, and the improved coverage and nucleosome position really means that you don't have to sequence as deeply to do what you would normally do with a Hi-C library. And so let's talk about that particular part of this assay. So if we look at the coverage profile, this is linear coverage, just loading the BAM into IGV and just looking at what bases are being covered between micro C and restriction enzyme-based Hi-C. What you can see here are the peaks where the nucleosomes occur. You can kind of see some gaps where there is maybe a nucleosome-free region, maybe a different protein is occupying that space. But what you also see in the Hi-C data is, yes, you see those enrichments around restriction sites, but you also see these big gaps, and those gaps occur from restriction enzyme motif deserts. There's no place for that restriction enzyme to go in and cut in these areas. So let's look at that in terms of binning, right? To make a contact matrix, we chunk the genome into, 1 kb, 4 kb, 10 kb buckets to try to look at how the coverage behaves and to capture those interactions between those bins. So if we look at a 200 base pair bin, which is suitable for micro-C data, you'd have to sequence quite a lot. But if we just look at a 200 base pair bin, we can see that almost every bin has good coverage. Even at places where there is lower coverage, it's not like it goes to zero. Now, when we compare that to Hi-C, we see this over-enrichment here, but we also see this complete lack of coverage here. So in order to get around this, typically what you do is you build bigger bins to try to rescue some of these fragments that don't have coverage into a bigger bin size. So let's do that. Let's make 1 kb bins. So now we look here and we see that across the board, the micro-C coverage, each bin has enough coverage to really be included into a contact matrix. Whereas the restriction enzyme-based data, you can rescue some of these lower coverage regions with areas that are over-enriched in sequence. But if you have too big of a region that doesn't have a restriction site, you're never really going to recover that bin in the contact matrix generation. And so the end result is that the contact matrices that you can generate with micro-C, these happen to be at 4 kb, look a lot more complete, partially because this coverage, and also partially because each bin has enough interactions to be able to capture these chromatin features. So not only are you getting more read support with loops that have been called, these are Raoul loops, but you can also start seeing these internal structures that just aren't being picked up in the restriction enzyme-based data. And really, in order to build a contact matrix that's really worth, identifying chromatin loops at, you would have to sequence the restriction enzyme data 35 to 50% more to kind of make up for this lack of read coverage. But that being said, these regions will always be left behind because there's just no place to incorporate the biotin in the library generation. So with that, I will hand it over to Miriam to tell you how you can make these libraries.
Hi, everyone, and thank you for joining us today. In this part of the presentation, I'd like to share with you what is involved in running the Dovetail micro-C assay in the lab. The micro-C assay can be completed in 2 days, and that is going from sample to a sequencing-ready library. It consists of 5 stages and a total of 18 steps. So day 1 is for cross-linking, digestion, and proximity ligation reactions. It includes stage 1, 2, and 3. At the end of stage 3, you would have generated proximity ligated DNA. This DNA is then converted into a sequencing library in day 2, and that would be stage 4 and 5, which take 4.5 hours to complete. There are several safe stops throughout the protocol for convenience, and there are also QC steps built into the workflow. Now, this is an overview of the workflow. In the next slide, I will walk you through the workflow stage by stage, and I will highlight the steps. These are very straightforward steps. They can be achieved using standard molecular biology techniques. Starting with stage 1. So in stage 1, the chromatin is fixed in the nucleus, then digested with MNAs. Following digestion, the cells are lysed with STS to extract the chromatin fragments. We use DSG and formaldehyde for cross-linking in a 2-step cross-linking strategy. So DSG has a longer spacer arm. It's a strong protein-to-protein cross-linker, and it allows to capture large protein complexes. So when you use DSG in conjunction with formaldehyde, we're increasing the long-range information captured in the library. I'd like to address a question of how we're rotating the cells during cross-linking. We just use a standard tube rotator with continuous rotation. An example is shown here. The Dovetail micro-C assay does not require sonication for library preparation. It's only relying on the enzymatic fragmentation or the digestion with MNAs. So achieving a good digestion profile is important to generate a high quality library. So high complexity and high enrichment of long-range information. To achieve optimal digestion, we recommend to freeze the cells before cross-linking. Also, the number of cells you're starting the assay with will influence the amount of MNAs used to achieve optimal digestion. In order to help with more accurate counting and to reduce cell clumping, we recommend that you harvest the cells at low speed spins and you use a swinging bucket rotor when harvesting the cells. We also recommend to count the cells before freezing. So you would harvest the cells, wash them with PBS, count, aliquot the number of cells you'll be starting the assay with, spin down to remove the supernatant, and then freeze the cells by just placing the pellet at -80 for at least 30 minutes before cross-linking and digestion. The standard protocol calls for 1 million cells, but if this number of cells is not available to you, we also provide digestion guidelines for lower input down to 10,000 cells. Moving on to stage 2. In this stage, this is a QC step that is included early on in the protocol to allow you to confirm that the chromatin is properly digested before moving forward. It will also allow you to quantify the total lysate or the chromatin yield that you're recovering from your cell sample. So how do you do that? How do you confirm that the chromatin is properly digested? It's by following these simple steps. You will first take an aliquot of the total lysate that you recovered after cell lysis at the end of stage 1. The remaining lysate will be stored back at -80. This is the lysate that you will use to proceed further. This aliquot is essentially cross-linked chromatin. So the first thing we'll do is reverse the cross-links, degrade the proteins with proteinase K, purify the DNA, and then we will run the DNA on an analysis instrument. You can use tape station, bioanalyzer, or fragment analyzer to assess a digestion profile. So looking at this digestion profile, this is analyzed by a tape station. The first peak here represents mononucleosomes. Here are the dinucleosomes. This is the trinucleosomes. The profile we're after has 40 to 70% mononucleosomes. So how do you determine the percentage of mononucleosomes in the digestion profile? You'll create a region that includes that first peak, which represents the mononucleosomes. And a tape station, usually this peak is between 50 and 250 base pair. When you create this region, a region table will automatically be generated under the electropherogram, and it will output the percentage of total, meaning how much of your DNA is within this region, 50 to 250 base pair. This is the percentage of mononucleosome. So in this case, the profile shows a 52.22% mononucleosomes, and it passes QC. This is another digestion profile analyzed on a fragment analyzer. Again, this is the mononucleosome. Here are the dinucleosomes, and here are the trinucleosomes. Based on the smear analysis, the mononucleosomes account for 48.4% of total. So this is also a digestion profile that passes QC. If you have a suboptimal digestion, we provide guidelines to allow you to adjust. So in the case that the chromatin is underdigested, and in this case, the mononucleosomes will be less than 40%, or if the chromatin is overdigested, so in this case, the mononucleosomes are more than 70%. Now, now that you've confirmed that the chromatin is properly digested and you've quantified the amount of lysate that you recovered by just quantifying the purified DNA on a qubit fluorometer, you can proceed to stage 3. So you'll start stage 3 with 1,000 nanogram of the lysate that you had stored at -80. If you have more than 1,000 nanogram, you can store the lysate, the remaining lysate back at -80. If you have less, you will use all of the lysate that you have recovered. The first step in stage 3 is to bind the chromatin fragment to chromatin capture beads. So the proximity ligation reactions will be carried out on the beads. Then you will repair the ends of the chromatin fragments. A custom biotin tagged adapter or bridge will be ligated to the repaired ends of the chromatin fragments. And then fragments that are co-localized in the 3D space are ligated to each other through that bridge to generate the proximity ligated chimeric DNA molecules. Following ligation, the crosslinks are reversed, the proteins are degraded, and the DNA is purified. This is a simple purification with bead cleanup. When starting the assay with 1,000 nanogram, we expect to recover around 400 nanogram at the end of stage 3. You'll only use 150 nanogram of this DNA for library preparation, so the final 2 stages. That means you will have more DNA to support additional library generation. This comes in handy if you would like to prepare technical replicates to include in the project, or if your project calls for higher depth, so you would need to generate more libraries. In that case, you can generate the libraries from this DNA, and you don't have to run the assay from the start. The intra-aggregate ligation reaction and the crosslink reversal reaction are incubated for 1 hour, but you can leave these reactions incubated overnight for convenience. All of these reactions are also incubated in a thermal mixer, so with shaking. Here's an example of the thermal mixer we use in-house. It's a standard mixer which has heating and cooling function. There is really no specialized equipment that you need in order to run the micro-C assay in the lab. The final 2 stages, stage 4 and 5, now you're converting the DNA that you purified at the end of stage 3 into sequenceable molecules. So these steps are just standard NGS library preparation steps. You repair the ends, add an adapter, and then purify the adapter ligated DNA. As I mentioned, before you start the library preparation, there is no sonication step. It's only the enzymatic fragmentation with MNAs that was at stage 1. That's the only fragmentation that is needed for library preparation. So now that you have purified the adapter ligated DNA, you can proceed to the final stage. Before the PCR amplification, we include a step of streptavidin bead pull down. So we're going to be pulling down and enriching specifically for the biotin-related proximity ligated DNA molecules. When these molecules or the template DNA is bound to the beads, we're going to set up the PCR amplification on the beads. And after amplification, we will clean the library and size select, then run this library on a fragment analyzer, bioanalyzer, and a tape station to check that the range is as expected. So we expect the library to be between 300 base pair, 350 base pair to 1,000 base pair. This library now is ready for paired end sequencing on any Illumina sequencing platform. I wanted to add that if the protocol as written is specific for Dovetail library prep module and primer sets. And that is, these 2 modules are offered as add-on modules for a more streamlined workflow. If you have a preference of a third-party library preparation kit that you routinely use in the lab, please reach out to us. We will let you know if it's compatible with the assay and how you can integrate it in the workflow. Before deep sequencing, what we recommend is to shallow sequence the library. So we shallow sequence the library between 1 to 2 million read pairs, and then we run a QC analysis to assess the quality of the library prior to deep sequencing. With this, I'll hand it over back to Corey to walk us through the post-sequencing QC analysis process.
All right, so you've sequenced the data. Now, what do you do with it? And I'm going to walk you through the steps and what you do after you get your data back off the sequencer. So it's good to keep in mind really what the core workflow is that your data is going to follow. So you're going to start with FASTQs. You're going to align with BWA-MEM to generate your BAM file. From there, we feed this alignment, we pipe this alignment directly into pair tools. Now, if you're unfamiliar with pairs or pair tools or things like that, the thing that you, I guess, want to keep in mind here is all it is it's a very simple table that distills your alignment information down to the read name, the chromosome of read 1 that it maps to, the midpoint, not the range, just the midpoint of read 1, the chromosome that read 2 maps to, the midpoint, the strandedness, and then the gives it a high C qualifier based on whether or not it's a PCR dupe, and then the insert size and the strandedness of that of the high C molecule. But regardless, all you need to keep in mind here is that it's just a simple way to reduce the alignment information to a much smaller data format that you can then convert into a contact matrix. Now, this is a very core workflow. There's many tools out there that kind of mask this data processing in a single executable to run it all. But today we want to walk you through exactly how you process these data to kind of really help you understand better what it is that you're doing. And I also want to point out here that we're following 4D nucleome standards. We're not changing anything. There's no magic that's going on. And as you can see here in the next couple slides, that it's actually pretty straightforward in how we process these data. So speaking of the process, it's the overview of it is actually pretty simple. We start by aligning and filtering low MapQ reads, unmapped reads, and PCR duplicates. And then we start classifying read pairs as cis or trans and characterizing the insert size. Sorry, it's late in the day here. And then identifying what are considered valid pairs. We're going to talk about that in just a second. And then if you shallow sequence, you want to know how complex that library is, so you know how deeply you can sequence it before you start throwing away sequence space on redundant molecules. And then the way that we do it, we spit out this report here, and I'll walk you through what that means for each step, and then we have a graphical representation of the data output. So before we start diving into each step of the process, let's take a second to talk about valid read pairs when it comes to high-C data. Now, if you've done restriction enzyme-based high-C data in the past, what you're looking for is that your insert size or your fragmentation occurs at a restriction site, and that the insert size captured by the read pair is longer than the molecule. So let's talk through what that means here. So if we look at what the process of making a high-C library is, it starts with fixed chromatin. If you're using a restriction enzyme, it's digested at restriction sites here designated by the stars. If you're using something like MNase or DNase to fragment where you don't have a motif, it's going to fragment randomly. And here we have the color kind of smearing across to show that it's more of a continuous molecule rather than with discrete stop points. Then we perform ligation, right? We perform that key step of proximity ligation. And the possible events that can occur is you can have a self-ligating event where you have this molecule here that kind of forms a circle with itself and just makes a circular molecule, right? That's known as a self-ligating event. Then you can have a religation event where one or two molecules cut and then ligate right back to the same site that they just got cut from. Or you could have what is considered a valid ligation event, where the ligation event spans a distance that's larger than your library molecule. Typically 1 kb is a good cutoff for that. Then what happens is if you're using restriction enzyme based, you shear to make sure that all of your molecules are in the sequenceable size range to put on an Illumina instrument. Now, it's a big thing to note here is that because we don't shear, the molecules are already small enough from the MNase digestion that there's no need to shear. All of your molecules are already within the right size to put on an Illumina instrument. Therefore, the self-ligation events aren't included into the library. Right? If it's a circle and you don't cut that circle anywhere, there's no place for you to adapt a sequencing adapter to. Therefore, it just never gets converted into a library, which is really nice. So by this method alone, we reduce a big part of the noise in the system. And then once you sequence, here are the possible read types. And really, how you classify these read types is whether or not in restriction enzyme space that your fragment occurs near a restriction site, and that the ligation event, that insert size, is bigger than your longest library fragment. So let's take a second to talk about that. So Miriam just showed you in the last slide how you visualize the molecule size of your final library, right? And so it's typically a histogram. It might not look as Bayesian as this, but it's a fairly normal distribution. And typically what you see is that you have the longest library fragment sits somewhere between 800 and 100 or 1000 bases. Now, if you were to sequence these molecules, right, and your insert size, like the physical distance that this molecule spans, is greater than the molecule that you've sequenced, the only way that that could physically happen is that if proximity ligation has occurred at a distance that's longer than the molecule, right? Religation events should all be captured in the insert size in between 200 to 1000 bases. Anything larger than that, the only physical way that can happen in a proximity ligation assay is if that physical distance that was being spanned was bigger than the molecule that you've sequenced. So that's how you go about classifying it. And so if you're using MNASE, right, with microC, really the only thing you have to consider here is that is the insert size bigger than your library fragment? And if it is, then it's considered a valid Hi-C read. The other thing to point out here is trans are typically considered valid read pairs. They might not be informative over cis topology like TADs and loops, but they are considered valid. You can use them for many types of applications, which we won't get into here, but the community as a whole considers them valid. All right, so let's talk about the process. And so we'll start with aligning the raw FASTQ data onto a reference genome. We use this using BWA-MEM. We use the -5SP flag. So what this is essentially saying is you're going to start reading from the 5' end of the molecule. And when BWA, when it sees a chimeric event, or in our case, when it starts reading either the bridge or when it starts reading a part of the genome that's too far away from the molecule, it stops and clips and then treats that as a single end read, and then takes the other part of the read and starts aligning from the 3' end as a supplementary alignment, right? And so then at the end of that, it merges that into one BAM file. So it's kind of like doing single end alignment, but with a single command instead of having to do read 1 and then read 2. So from there, we now have our reads mapped to the reference genome, and then we can filter out any unmapped reads and any low mapped Q reads. Here we have a cutoff of 60. And then we take these mapped reads and feed it in, we start piping it directly into pair tools. Now, pair tools, what it will do for us is it will flag PCR duplicates and remove those. And so what we then do is we ask how many reads fall into these categories of, first off, how many did we sequence? Here we're looking at 2 million. How many of those reads were unmapped? How many of them were mapped? How many of them were PCR duplicates, and how many of them are non-duplicate read pairs? And so we report those counts, and then we show them as a proportion or percentage of the total read pairs. So this bracket here says all of these fractions are calculated by simply taking your count and dividing it by the total read pairs that you sequenced. And here is just a graphical representation of that. So here's all the data, or here's all the raw reads. Here's what aligned with a good map Q, and then here is the no dupe read pairs versus the duplicated read pairs. It should be noted that as you sequence more, your PCR duplication rate is going to go up just because you're going to start capturing more duplication molecules with deeper and deeper sequencing. All right, so now we're going to take these no dupe read pairs, these high quality mapped read pairs, and now we're going to start classifying them in terms of their Hi-C-ness, right? What is their proximity ligation category? So first, we take those no deep read pairs, and this is something that Pair Tools does as a part of its data processing. And it asks, are they cis reads or are they trans reads? Cis meaning that they occur on the same chromosome, or trans meaning that they span different chromosomes. So, okay, now we have our cis and trans bucket. We know that trans are valid, so boom, we don't have to do anything there. But for cis, we want to understand, are they religation events or are they valid ligation events? So in order to do that, like I was mentioning earlier, it's all about understanding what that insert size is. And so what Pair Tools essentially does is it generates a histogram where it counts number of reads at different insert sizes, right? And then puts a cutoff where you kind of tell it to put a cutoff. Here we have 1 KB. And we just say, how many reads occur that are less than 1 KB here? How many reads that occur that are greater than 1 KB in this bucket here? And as you can kind of see, as you get farther and farther out, the count seems to be dropping, but the sum of that count is actually quite large, right? So the total number of counts here in this invalid space here is only about 115,000 read pairs, where in the tail of this curve here, we're getting, over 1 million of our read pairs are greater than 1 KB. And moreover, about 900,000 of them have an insert size that's greater than 10 KB. That's really informative over that topological space there. And so from there, now that we have our counts and we know what the insert size is for each one of these, we can just, pass the filter that says any cis read that's less than 1 KB is invalid. Anything that's greater than 1 KB is valid, and trans reads are valid. And here we report again the counts, and then we show them as a proportion of no dupe read pairs. Here we want to be thinking about the amount of usable data rather than the amount of total data that was generated off the sequencer. So that's how these percentages look. And here we're just giving you a graphical representation of, okay, here's your total no dupe read pairs. And then here are the trans reads, here are the cis reads. The trans can go straight into the valid bucket. And then the cis, we need to characterize the insert size. These are less than 1 KB, these are invalid, they don't get moved to the valid, and then anything greater than 1 KB is considered valid. All right, so now you have your proportion of valid read pairs. And so if you shallow sequence, the next thing you want to ask is, how deeply can I sequence these libraries? Because Hi-C applications are really about generating enough read pairs to populate your contact matrix. So we do this using a tool called Preseq. And essentially what it does is it takes your current sequencing depth and asks how many PCR duplicates are contained within that shallow sequence library. And then it kind of subsamples a little bit to get an understanding of how frequently they pop up, the PCR duplicates pop up in shallow and shallow sequencing, and then they use that to build a model and extrapolate that out as if you were to sequence the data really, really deeply. It is an estimate, so it's not 100% accurate, but it does give us a good idea of whether or not your libraries are complex. And so typically what we do here in this extrapolation curve is we draw a line at a Hi-seq lane. You could draw that line wherever you want to, but most people sequence around 30x with one library. So we want to understand what the behavior looks like at that 300 million read pair size range. And so you could either be quite complex, right? High complexity, looking at about between 225 and 250 million unique read pairs per 300 million read pairs. You could be okay or moderately complex here, looking at 100 million unique read pairs per 300 million. And if you're in this bucket, all you would need to do is go back to your proximity ligated DNA and make a new library pool and then sequence to be able to bring that complexity up. Or you could have really low complexity, and this is indicative of something failing in the library. So then we would recommend you going back and restarting the assay from scratch. So that's kind of how you would go about estimating that complexity. So now that you have your valid pairs, you've sequenced them to the depth that you want to, now what? Now it's all about contact matrix land, right? We build contact matrices. There are actually a handful of ways to do this. The two main ones are with juice tools and with cool tools. Now, Juicer builds those .hic files that play really nicely with Juicebox. If anybody's used that before, it's really user-friendly. And then Cooler, I think, is a little bit more dynamic, a little bit more flexible. The learning curve on it's a little bit steeper, getting things up onto high glass can be a little bit more complicated if you're not used to it. If you are, then it's not that big of a deal. So I'll show you how to do this in a second. And then once you're done generating contact matrices, you're on to your analyses. And we'll have a future webinar here in a few weeks that really does a deep dive into how do you really make the most use out of your micro-C data. So before I close out here, I want to show you all two things. First one is that we have some really helpful documentation available for everybody. So we have essentially, let me pull up a different screen here. We essentially have this entire webinar, or the second half of this webinar, I should say, captured in this really nice documentation to remind everybody what a valid read pair is, to what the workflow is, how we go about doing that, how we go about calculating the stats. It's essentially what I just told you, but in writing if you ever need a refresher. The next thing that we have, which we're really excited to share with everybody, if I can find my mouse, is this documentation that we've put together on Read the Docs. This is a really flexible platform for us to really help users understand what they're doing and really learn how to process these data. So we have, a little refresher on what micro-C is and what it can do and all that fun stuff. And then we tell you what tools you really need, where you would go to download them, right, and how to kind of get started. Then we have some pre-alignment steps, how you need to get your reference genome together to do the alignment, how do you need to prepare your chromosome size file to build your contact matrix. Then we provide you all of the steps to do the alignment and run the pair tools process. If you've gone through this before and don't want to read through everything and go step by step, or if you're really impatient, we do have everything piped together that will take you from your alignment to your pairs file and your alignment file, along with an example of how to do that. Or we break it apart step by step so you can run things one at a time and really see what the output of each thing is doing. I highly recommend everybody check this out. It's a really good tutorial. Next, we talk about library QC. We share our little Python script that does all of this counting for us, that basically summarizes the output from pair tools. And then we have a whole section here on generating contact matrices, right? So where you would go to download the Juice Tools jar, right? How to install it, how you would run it to actually build your contact matrix, and we tell you exactly what each flag in that command is doing. So there's no like, what does this XMX mean? It's actually how much memory do you flag to build your matrix, right? Which can be really important if you're using lots of data. So we've really kind of taken our time here and really broken this down step by step, so you can see exactly what output from each tool then feeds into the next input. So you really get a good sense of what your data is doing. We also have a workflow here for going from your pairs file into a cool file with Cooler. Again, it's actually pretty straightforward to get going on this. So the learning curve is actually pretty shallow if you go this route. The other thing I would like to point out here is we have data sets available for you all to just download and play with and test. We have a very shallow sequence data set at 2 million read pairs. So if you're just wanting to check to make sure the install for all the tools work and you can get through the pipeline in a really short amount of time, start here, make sure everything's working. And then if you're getting ready, right, or you expect you're getting between 800 and a billion reads coming off the sequencer in the next week or so, and you want to make sure you really know, what timelines to be expecting with that amount of data. We have an 800 million read paired data set for you to download. They are large files, so if you download them, be prepared that they're quite large, but you should grab them and start processing them so you have a feel of how to work with data that's that large. So with that, I am going to stop share the last screen here. And just again, call out that, these tools are out there for you to use. So please feel free to use them as much as you want to. And that we do have future webinars coming up, one being this sort of recapitulation of this with high chip data. And then we're going to spend the following two webinars about really digging into the data analysis. How do you use these data sets to call features, and then maybe to even think about start asking biological questions with these data.